Assalamu alaikum and good morning. Today I'm going to talk about viruses, their structure and replication. So in other words, we are going to go a little bit in depth to understand what are viruses. I think during the first lecture, there was something mentioned about the spectrum of illnesses and what are those agents that play a role in causing illness and injury. So this could be anything from environmental to genetic factors. In between them, there is something which relates to infections. And as a matter of fact, infections can be caused by so many factors. And already you've been, I think, involved in trying to know those different bacterial infections, for example, parasitic infections, and so on. And today we're going to talk about viruses and their infections which they cause altogether. So the objectives of this lecture here is that we should be able to really know something about the history of the discovery of viruses. And then we should know what is the definition or how the definition of viruses have developed. And then we will be talking about the virus particle and how these components inside the virus particle are organized and what sort of shapes do we see among viruses. Other than that, we're going to talk about replication, and we will define what is replication in the case of viruses. And also we will be talking about some of those the effect of those viruses on cell and what happens. So as you can see in this diagram, there is a ruler which is divided into different parts. It starts with 1 centimeter and it goes up to 1 angstrom. And on top of this ruler, there is the different agents which we wanted really to demonstrate what is the relative size of those with this ruler. And as you can see, viruses usually fall in the category of 10 to the minus 7. And this is usually even less than 1 micron, which usually the bacteria tend to be within the range of 10 to the minus 6 or 1 micron. Now, as you can see below the ruler, there are different methods of trying to see what are those those different agents. And as you can see, the spectrum of light microscopy usually falls between 1 millimeter and 1 micron, or a little bit more. However, electron microscope would be even far better, where you can resolve even up to 10 to the minus 8 in certain cases, or up to probably nearly 1 nanometer. With X-ray or X-ray crystallography, you will be able to really detect those different proteins and atoms, while the nuclear magnetic resonance will help you even identify further all those tiny molecules. Now, as I said, you can see that viruses usually falls within the category of electron microscopy, meaning that most viruses, except for pox virus, could not be seen by a light microscope, while the majority of viruses can be detected by electron microscope. Pox virus usually tends to have a structure which is quite large. Therefore, light microscopes really helps in detecting pox virus. And this is probably the only virus that could be detected by a light microscope. Now, again, this is just to give you an idea of the size of viruses, which we're going to talk about, and a relative size of a bacterium, Staphylococcus aureus. So if we assume that Staphylococcus aureus size is a 1 micron, then you can see what are the different sizes, for example, of herpes, influenza, and picorna. So they would be very, very tiny particles in comparison to a Staph aureus particle. Now, among viruses, again, the sizes of different viruses vary. Some viruses have very large particles, and some have very small, tiny particles. Large, as example, herpes simplex, those are one of the largest virus families, while parvoviruses are tiny viruses. And you can see even in this picture the size of the molecule of IgG and the size of the virus, which is relatively a little bit larger than that. However, in between, usually all those viruses tend to lie in their sizes. Now, what is the word virus? Now, the word virus is a Latin word, and it stands for poisonous fluid. Having said so, the viruses themselves were not been or have not been able to be detected until the invention of the electron microscope. The infections usually they cause were known since early civilization. We know that viruses were there, however, we could not see them until the invention of the electron microscope. And between the 16th and 19th century, many studies demonstrated the presence of viruses and acknowledged that viruses are within us. And probably the most important work of Ivanovsky and Bejring showed that infections with tobacco mosaic virus, which is a virus that infects plants, they were able to demonstrate that this virus could be transmitted from one plant to another. As a matter of fact, during the late 19th century, tobacco was considered a commodity. So the tobacco was very expensive at that time. And it used to be brought from the East to Europe. And during the market and selling, some scientists were able to see that there were leaves of tobacco that looked normal. However, other leaves, they had some sort of a mosaic structure on the leaf. And this really affected the price of the leaves, because they thought that this mosaic effect is usually a disease which is really affecting the tobacco plant. And as I said, Novisky and Bejring were able to demonstrate that by taking a leaf or an infected leaf, which showed this mosaic pattern, and they squeezed that and they got all the extracts or the sap, and they took that extract and placed it on top of a normal leaf of tobacco. And they rubbed it with some material so that the sap or the extract would enter into the leaf. Now, what happened is that after a few days, they found that this leaf started to get the same mosaic pattern that, like the one they started by taking the extract from. And hence the name poisonous fluid came, or virus came into usage, and this was considered the beginning of the discovery of viruses and their ability to be transmitted. The same thing happened with some animals at that time. You see, some of the cattle were infected with what we call foot and mouth disease virus, and those usually had their face all filled with blisters and vesicles, and their feet at the same time had also similar thing. And taking some of these solutions on the face and placing them on a healthy cattle or cow led to the transmission of the similar characteristics of disease that was present in those cattle which we started with. So this was again was considered as a poisonous fluid that had the ability to transmit viruses from those infected to those which are non-infected plants and animals. Now, at that time, probably one among the first definitions of viruses is that they were very small, tiny, and they were filterable. So you could filter them, meaning that the sap or the extract which was taken, even when filtered, still the filtered solution contained those agents or particles. And also they could not be grown on media that was used for bacteria. You all know that bacteria can be grown on artificial media, as we do in the lab. Now, the same thing when we tried really to cultivate viruses or this poisonous fluid, we were not able to get any results. So, therefore, small filterable agents and could not be grown. Probably this was the first definition of viruses. However, after a few years, things started to develop. And of course, when cell culture techniques were introduced, the electron microscope in the early 20s of last century, and by the 50s, when molecular biology studies started to expand and our understanding of the nucleic acid and DNA appeared, all this helped in identifying viruses and place them in a very distinctive group among the biological objects. And therefore, we accordingly, those had certain structures and they had the ability to replicate. As a matter of fact, some people might use multiplication, but multiplication is not a right word. So please be aware that viruses do not multiply, they replicate. Now, when we come to look at viruses and compare them to bacteria, for example, we find that viruses usually, as I said earlier, do not grow on artificial media, while bacteria can grow on artificial. Bacteria can undergo division by binary fission, and this is not observed with the viruses. Now, bacteria usually have both DNA and RNA, as any organism. However, in viruses, you cannot have both. It's either DNA or the RNA that is present. Now, in bacteria, we know that bacteria are very complicated organisms. They have ribosomes. In the case of viruses, we don't see ribosomes. However, in certain cases, ribosomes may be present, but those viruses which contain ribosomes have acquired them from the host or the cell. And for example, viruses, we know that they are not sensitive to antibiotics in comparison to bacteria. Whenever you get a bacterial infection and you go to the doctor, he will advise you to take antibiotics, which has a big impact on destroying bacteria and controlling the disease. However, this is not the case with viruses. So simply, antibiotics do not work on viruses. And mind you, also, for example, muramic acid, we tend to see that this is present in the wall of bacteria. However, this is not present in the case of viruses. Now, having said so, viruses usually have certain components. As you can see, this is a picture or an electron micrograph of a virus, meaning that this has been taken by an electron microscope. The picture has been taken by an electron microscope. And as you can see, that the virus, the whole virus is called the virion. The whole virus is called the virion. Now, inside the virion, usually there is what we call the capsid. And the capsid, as you can see here, is a spherical or circular component. And if you look carefully, you will see that this capsid is made of tiny units, which we refer to as capsomeres. So capsomeres form, in total, form the capsid. Now, one of the things which we cannot see here in this micrograph is the nucleic acid, because simply it is hidden inside the capsid. And usually the nucleic acid plus the capsomeres, we refer to them as the nucleocapsid. But one thing to remember is that these capsomeres, which we just mentioned, are made of proteins. So those proteins usually aggregate to form a certain structure that helps in protecting the nucleic acid inside it. So it helps in protecting, and it helps in transferring the nucleic acid from one organism to another. If you look carefully in this electron micrograph, you will see that the virus is surrounded by what we call the envelope. The envelope is a lipid bilayer and usually is acquired from the host. Not all viruses have an envelope. Some are naked and they just have a capsid on its own, made of capsomeres. And some viruses, they tend to have the envelope. You can see here, for example, in the in the left corner, we have a protein which, after folding, tends to take a certain structure. And you know, because proteins usually they have a primary structure, secondary structure, and tertiary structure. So by the time they start to form a tertiary structure, those proteins, they usually are asymmetrical. When they come together to form subunits, they start to become symmetrical. So the protein on its own is asymmetrical. And the more foldings you get, the shape of the protein usually starts to vary. Now, in the case of the more than proteins coming together and forming a symmetrical structure, which here in this case is going to be the sphere or the circular structure. Now, what do viruses lack? After we talked about viruses and the components which they have, what do they lack? Obviously, there are so many things that are missing in viruses. For example, there is no mitochondria in the case of viruses. And this is a machinery which is used to generate energy. And also, there are so many tiny structures which are not present in a small virus. So altogether, what we can come to, or the conclusion for a definition of viruses is that those viruses usually are obligate intracellular parasites. Intracellular parasites, meaning that they require a host to parasite on and depend on their machinery to make new virus particles. We're not going to go into the philosophical definitions of whether those viruses or the virus particles are live or not live. It all depends on how we define life, really, and we don't want to go into that in depth. So mainly, I think probably at this stage, it's better to stick to this definition where viruses are obligate intracellular parasites. Now, as I said earlier, the viral components are made of different components such as nucleic capsid, which is the protein coating or surrounding the genome. The genome is the nucleic acid. So there's a protein which surrounds the genome. And the capsid usually is the outer protein shell. We said that the envelope is the lipid outer membrane, which is usually derived from the cell membranes. And in some viruses, there are proteins which are made by the virus and inserted into these lipids during the process of making new virus particles. And usually we refer to those proteins as glycoproteins, such as the hemagglutinin of influenza and other protruding proteins. Usually those proteins which are protruding from the envelope, usually they are a good primary target for neutralizing antibodies, which help in making vaccines against viruses. And we mentioned that the genome in the case of viruses is either one of the two, either DNA or RNA, however, never both. Now, what we have seen, we talked about the structure with the nucleic acid and the components. What we have seen is that those viruses usually tend to form either one of these symmetries. The symmetry which we observe usually is the cubical or spherical. Most animal viruses are cubical or spherical. However, in certain cases, you can see that the virus structure is usually helical, just like the DNA, which is made of a helix. And as a matter of fact, here you will see that the nucleic acid is surrounded by protein molecules, and those tend to really be packed in such a way that they protect the nucleic acid from external effects. The same thing with the spherical virus particles. You will see that those small proteins, the subunits which we mentioned earlier, usually are located in such a way that they sit within triangles. The more the triangles you have, the more spherical the structure is. And you can see that this probably the spherical structure is one of the best structures that requires minimum amount of energy really to form this structure and protect the virus particles and again stay in a very solid state. This is one of the buildings here in Muscat. I wonder if you've seen this building. You see on the top there is a dome. And if you look carefully, the dome is made of triangles. So this is precisely what I've been talking about. The more triangles you have, the spherical structure would become. And that's precisely how the proteins usually sit within the triangles to form this spherical structure. Now, there are certain exceptions where we tend to see that some viruses may have both the spherical structure and the helical structure. And probably the best representative of such viruses is what we call the bacteriophages. Now, the bacteriophages usually are the viruses which tend to infect bacteria. I probably did not mention that earlier, but I want to say that every living organism in the world could be infected with viruses, and hence bacteria are usually infected with bacteriophages. Now, as you can see here, the spherical structure forms the head, and then there is the helical structure, which ends with the tail, and the tail usually contains very sharp structures. As you can see here at the end, these structures resemble knives-like structure. And as a matter of fact, they are utilized for that purpose, really, to penetrate into the bacterial cell wall, and then this will help in injecting the nucleic acid which is present in the head through the helical structure and into the host bacterium. Now, we said the genome is the nucleic acid. And what we have seen at the beginning, we saw that there are viruses which have very large structure, while some viruses have very tiny structures. Now, those viruses which have large structures usually are complicated, and they tend to have a large number of genes, high number of genes. And while in comparison, the small viruses usually have a limited number of genes that usually make a very small number of proteins. Now, those viruses with large complicated genes usually make so many proteins, which helps in replication and so on. Now, we said that the genome of the virus could be either DNA or RNA. Of course, there is a uniqueness among viruses, is that the DNA in certain cases could be single strand. You know, the DNA is usually a deoxyribonucleic acid and is made of 2 strands. However, in the case of viruses, you could see DNA made of 1 strand. And the RNA, as a matter of fact, we know that the RNA is made of a single strand, but in the case of viruses, we can see that some viruses contain double-strand RNA. Now, this DNA or RNA usually is linear, meaning that it has open ends. While in some of the genomic structures, there is what we call circular genomes, meaning that the ends of the molecule usually come together to form a circle, a full circle. Now, what we have seen also is that some of those viruses, they have either intact, meaning that the nucleic acid is one piece, or segmented, meaning that the nucleic acid is made of segments. Each segment usually would contain different genes that tend to provide different proteins for the virus. Now, one of the things which I really advise everyone is that nucleic acid can be very infectious. So when handling viruses, please be careful when you handle them, because even when the nucleic acid is naked without the capsid, still, if there is an opportunity for that naked nucleic acid to enter through a break in the skin, then this will cause an infection. So you have to really be very careful when handling viruses. Now, one of the things which we know is that viruses usually cause diseases, and in order for them to cause diseases, they ought to really be able to enter into our systems. Now, for that purpose, there are different ways for the viruses to enter into our body. Among them is the inhalation of the virus particle, meaning that the virus particle goes through the respiratory system, or by ingestion, meaning that you eat some food which is contaminated or drink water which is contaminated, and this goes through the alimentary tract and further on to other parts of the body. Inoculation, as you know, inoculation is done by using either components of a virus or by using the same virus itself. Could be killed or it could be life attenuated. We will talk about that in a later lecture. Among the other ways of entry into the body is by congenital transmission, meaning that the virus is transmitted from the mother to the fetus during the pregnancy stages. Now, this, as we said, leads to the entry of the virus into our body. So what happens when our body detects those viruses? Usually there are different ways of defense mechanisms. Among them probably is the non-specific defense mechanisms, and probably the best example is the skin. You know, the skin is a protective layer which prevents viruses from entering into the body. However, if there are breaks or cuts, then a high possibility that the virus can go through these cuts or breaks into the body. Now, among the other defense mechanisms is the specific defense mechanisms. Probably the best example is the immunological behavior. You know, once those virus particles enter into the host, the host will respond by through the immune system by antibodies that can fight the virus and so on. This, as a matter of fact, this part, my colleagues, the immunologists, will be talking in more depth to make it clear to you what are the specific defense mechanisms that are utilized by the body during the process of infection. Now, as I said earlier, viruses do not multiply, they replicate. And for that, they ought to really go into different stages so that they can make new virus particles. So one virus particle could make hundreds of virus particles. How is this achieved? This is done through different stages during the process of infection. The first stage, usually the virus particle is adsorbed to the cell surface. The target cell, the virus usually comes to this target cell and gets absorbed into the receptors which are present on the surface. Once the virus attaches to the cell, the other process starts, and that is the entry of the virus particle into the cell. This is achieved by different methods. We'll talk about them in a second. Now, other than that, once the virus particle enters into the host or the cell, the other process starts, and this process is referred to as the uncoating, meaning that those proteins which form the capsid starts to break to release the nucleic acid into the host. Now, once the nucleic acid enters into the host, further process is achieved or done through process of transcription and making new proteins or synthesizing of different virus components. And also the proteins which are being made, some of them are structural, some of them are non-structural or enzymatic, and those will help in making large amounts of nucleic acid, new nucleic acid, and together those new nucleic acids with the virus components or structural components that are being made, they will come together to assemble a virus particle which again is released either by rupturing the cell or the host, or through budding through the host and acquiring part of the envelope of the host into their structure. Now, as I said earlier, the viruses usually attaches to the cell surface, and there are ways of attaching. So there are signals, receptors that signals for the virus particle that helps in the virus moving towards those receptors. And once the virus attaches to the receptors, then further on there is another stage which is going to be followed by the entry of the virus particle into the cytoplasm of the host, of course by penetrating the lipid bilayer. As you can see in this diagram, there are different ways of entry. Now, first of all, there is what we call the surface fusion, where the virus fuses with the surface of the host, simply because both the surface of the host and the virus particle are made of the same proteins or envelopes, where they can fuse together easily. Once they fuse, this will help in releasing the capsid into the host, and then after a process, there is what we call the direct uncoating, which leads to the nucleic acid being released from the capsid. Now, other than that, there is what we call receptor-mediated endocytosis, where usually the virus particle fuses in an endosome. As you know that the host usually tries to protect itself, so this leads the process to form an endosome that is taken inside into the host, where usually there is an attempt to break down those foreign particles. However, viruses, usually what happens is that they fuse within the endosome. Once they fuse with the endosome, that means that the capsid is left free to enter into the host, and after that releasing the nucleic acid by the process of uncoating. Other than that, there is also a process where lysis of the endosome. So the endosome engulfs the virus particle and takes it inside, and after that the virus particle, as we said, lyses the endosome, and the virus capsid or capsomeres enter into the host. This is again a diagram which really gives you an idea how this process or the stages, different stages of virus infection are achieved. So we said at the beginning there is the virus particle, which is adsorbed to the cell surface, and once it's adsorbed to the cell surface, it starts the process of entry. Now the entry or penetration leads to the virus particle to get inside the host, and as I said earlier, this virus particle is going to be released, and therefore releasing after a while by the process of uncoating, releasing the nucleic acid. So the nucleic acid is going to be present inside the host. Now what happens here is that this nucleic acid is going to be transcribed, meaning that there are messenger RNA which is going to be made from the nucleic acid itself. Some of those proteins which are going to be made, usually, as I said earlier, they're going to be structural and non-structural. Some of those are going to be inserted into the lipid bilayer or the envelope of the host, as you can see these green dots. Now, in addition, there are proteins which are going to be made and those we said are structural proteins, while the non-structural proteins are going to go back and work on the nucleic acid. Once it works on the enzymes, works on the nucleic acid, it's going to make so many copies of new nucleic acid. And this is what we refer to as the replication process, where new nucleic acid is going to be made. These molecules of the new nucleic acid, which are similar to the original nucleic acid, are going to meet the virions, those empty particles which do not have any nucleic acid. And once they meet each other, they assemble. So they come together to form a new virus particle, which during the process tries to mature by moving to certain areas within the envelope that contains the nucleic acid. This will help the virus particle to move and acquire the envelope together with those proteins that have been inserted. So this is just a viral life cycle. We know that, as I said earlier, that viruses usually are made of DNA or RNA, and those that are made of DNA, some of them are complicated, so they have their own enzymes, while some of the host or some of the viruses which are so small depend on the host cell polymerase. So viruses have either their own polymerase or they depend on the host polymerase for the purpose of replication. Now, we have seen that viruses can replicate, and they replicate in different ways. For example, DNA viruses undergo what we call rolling circle mechanism, while in other viruses they can undergo bidirectional replication, and there is what we call linear replication, as in the case of adenovirus, or RNA intermediates, as in the case of hepatitis. For the rolling circle, as we said earlier, some of those viruses have linear nucleic acid. They are circularized, so the both ends come together, and then one of the enzymes sits on this double-strand nucleic acid and makes breaks, and it starts going around in circle. And as it goes around in circle, it tends to read the strand and makes a new strand. Therefore, there's going to be a continuous strand to a very large size of nucleic acid head to tail. So they are all connected. And then enzymes, certain enzymes, specific enzymes, cleave or cut in certain regions, making a unit length molecule. This unit length molecule is going to be packaged, going to be taken and packaged into a capsid, and then the mature virion will appear. As I said, in the case of bidirectional DNA replication, as in the case of papilloma, they are circular nucleic acid and they have the origin of replication. Then the two strands separate from each other. And at the same time, there is an enzyme going to read each strand, separated strand, in the opposite direction. So while they are moving, they are going to make a second strand, and at the end of their movement, they are going to form two separate molecules. Those separate molecules are going to be cleaved at certain areas or regions. For RNA, RNA also replicates in a different way. It all depends on how those viruses, RNA viruses, are made of. You see, initially when we talk about DNA, we talk about two strands. Now, if we consider one strand as a positive strand, then obviously the other strand, which is complementary to the first strand, is going to be a negative strand. Now, usually messenger RNA is going to have a positive strand simply because it's made from the negative strand. So the positive RNA, in that case, some of the viruses have a positive, similar to the messenger RNA. However, other viruses may have a negative strand RNA. These are, by the way, not charges, remember. It's just a way of explaining how those nucleic acid are complementary to each other and so on. All viruses usually replicate in the cytoplasm except influenza and so on. Now, as I said earlier, once the virus enters into the cell, there is the effect, which is, or the host response, which is going to be mentioned in another lecture. And once the virus replicates and make new virus particles, what's going to happen? Now, what we have seen is that there are one of these three effects of virus infection on the cell. Either the cell is lysed and death occurs due to the cytopathic effect, as in the case of poliovirus, where those usually destroy the anterior horn cells, or in the respiratory cells, where they tend to kill the respiratory epithelium, or there is very minimal damage, as in the case of hepatitis, which does not cause any pathological harm to the liver, but the immune response in this case would lead to the damage of the cells. Now, the third effect of viruses is what we call transformation, meaning that the cells which have been infected by a virus is transformed from a normal cell to an abnormal cell, i.e., it becomes a cancer cell. This is noticed with many viruses such as human papilloma and genital cancer, hepatitis B and hepatocellular carcinoma, EBV or Epstein-Barr and Burkitt's lymphoma and nasopharyngeal carcinoma, and human T-cell leukemia virus. All these viruses has the capability of transforming cells and making them as cancer cells. So up to here, I'm going to stop. If you have any queries or questions, please don't hesitate to write to me or ask me if I'm available in my office about your queries. Thank you very much.